Welcome back to the playlist on ketone bodies. In the past few videos, we've been looking at ketone body biosynthesis, ketone body catabolism, and we've tried to overall get an understanding of why ketone bodies are important. Now, we went into the biosynthesis of all three of the ketone bodies, which include acetone, beta-hydroxybutyrate, and acetoacetate. But what textbooks tend to do is they tend to focus on the catabolic pathways for acetoacetate and beta-hydroxybutyrate. Okay, but one of the things we're going to focus on in this video is something that's recently come into understanding, at least in mammals. And we're going to look at the catabolism of this guy right here. This molecule right here has a special name. It's called acetone. So this is called acetone. This is one of the ketone bodies that was once thought to sort of be static. It just got excreted, and we didn't really understand the pathway very much. One thing about acetone that's interesting is that in a diabetic, when they're severely running on ketone bodies, the breath smell that they get is actually exhaled acetone. So you can actually detect when somebody's in ketoacidosis, you'll smell the acetone on their breath. And they've actually recently started to come out with this pathway that describes acetone degradation. So it is something that you can obtain energy from, and what we're going to find is that it actually gets degraded to pyruvate. So that's just sort of an introduction on this video. And I just want to sort of hammer into your heads that ketone body synthesis is not just about starvation. It's not just about diabetes. Ketone bodies are an incredibly important source of energy. Imagine that you're eating a very, very low carbohydrate diet. It's filled with proteins, which ultimately get degraded to amino acids and lipids. Well, the lipids will undergo beta oxidation, and you'll end up producing acetyl-CoA. So when lipids get beta oxidized, and we'll run into that when we get to the beta oxidation playlist, is that the end product for each round of beta oxidation, except for the last one, is one acetyl-CoA. The last one yields two acetyl-CoAs. But the acetyl-CoA is not just used by the liver cell that makes it. It's actually turned into these three ketone bodies, and the ketone bodies get dumped into the blood, and then they go to peripheral tissues. And then this is one of the processes that occurs, is you'll have acetoacetate conversion to acetone, and then acetone will get metabolized into an energy-producing pathway. And so we have to keep in mind that ketone bodies are not just a starvation type of thing. If you're eating a high-protein diet and not a lot else, you will be running on a lot of ketone bodies. Your liver will be very active in making these molecules. And it's not like you can't run on them. In fact, for example, I run on a very, very high-protein diet, very high-lipid diet, and extremely low-carbohydrate diet. And there's absolutely no problem brain-wise, there's no problem at all running on ketone bodies. If there was a problem running on ketone bodies, you have things like lions, they wouldn't be able to run after their prey. I mean, these things are so obvious and they're out there. If animals could not run on these things, they'd be dead, because that's all they eat is meat. They're not getting any carbohydrates. So clearly, there has to be some way to run on these things, and it turns out it's actually very easy. Your liver makes these ketone bodies when you eat a high-protein diet and high-lipid diet, and then the ketone bodies are dumped into the blood, and they go to serve peripheral tissues. And what we're going to do now, is one of the processes that occurs. So this is going to be the video on the degradation pathway for acetone. But what we have to do first is we have to get some preliminaries out of the way. We have to talk about this molecule right here, and this molecule is acetoacetate. Now, acetoacetate is one of the three ketone bodies, and specifically you can actually physically see the ketone right here. So here's the ketone right here of acetoacetate. And it could go through equilibrium with beta-hydroxybutyrate through an enzyme. But acetoacetate can directly get degraded, or it can get converted to acetone. Now, here's one thing that I want to mention. There's a lot of things in this pathway that are just coming into fruition, and they're not well understood. Now, with acetoacetate decarboxylase, this is not actually known to exist in mammals. certainly what happens is in lower level animals like prokaryotes, this enzyme is known to exist. But in humans, it has not yet been isolated, which is really unusual because you normally think an important pathway like this would have it isolated, but it turns out that acetoacetate decarboxylase has not been isolated in mammals. Okay, it's very possible that this reaction occurs spontaneously in humans, and it's very possible that that is the case. In fact, we can actually look at a pseudo mechanism for this. It's actually really easy, assuming that you have some proton here in solution that can just come from water, right? What could happen is these electrons on this oxygen could kick in here, and that would form carbon dioxide, and this bond right here would break, and it would come out and abstract the proton, and you'd get acetone. And in the process, you'd get this carbon dioxide. So this enzyme has not been isolated in mammals, so there's thought that this process might occur spontaneously. Let's write that down. This process might be spontaneous. Now, obviously, in biochemistry, there are many reactions that have a negative delta G. So when you're being really technical and you say something spontaneous, what you're talking about is it has a negative delta G. But oftentimes when you're talking about enzymatic pathways and you say a process is spontaneous, what it means is it does have a negative delta G, but you're saying it's non-enzymatic. So we're going to qualify this by saying that it's possibly non-enzymatic. Okay, that this reaction does not require an enzyme, that it's very simply just a decarboxylation that occurs in solution. Now, not all of the acetoacetate would do this. It's a probabilistic sort of thing. Are you in the right chemical environment for this to occur? And there's probably a rate that's associated with it. Okay, so not all of the acetoacetate makes it into acetone, but some of it does. Okay, so let's say that in prokaryotes, acetoacetate decarboxylase does exist. It's known to, but in humans, it's very likely that this enzyme does not exist and that the process is purely spontaneous and non-enzymatic. But in any case, we get acetone, which is shown here now on the left side. Now, there's another molecule down here, and this is called isopropyl alcohol, or sometimes they just shorten it to isopropanol. Isopropanol. And isopropanol is not something that you would, ingest every day, okay? But if you're working in a laboratory setting, when you're in the chemistry lab and you're cleaning stuff, you're using isopropyl alcohol, okay? So there's always a chance that this stuff could be inhaled. And if it's inhaled, it's going to have to be catabolized. So it's not like you go out and get some food and this is in it. No, this is probably not going to be in your food. The amount that's in there is going to be absolutely negligible if there's none at all. Okay, but in any case, if you're in a lab, you're going to probably inhale some of this. But it's going to get converted into acetone by this enzyme, isopropanol dehydrogenase. And specifically, this is an NAD-dependent enzyme. So you'll have NAD right here. It's going to come in here and generate NADH. Okay, this is not like a huge contribution to your overall energy, because in any case you really shouldn't be inhaling a lot of isopropanol, but it does happen, and if it does, it gets converted to acetone. Okay, so now that we've got that out of the way, we're at acetone. Let's talk about its catabolic pathway. Well, it turns out that acetone begins its catabolic pathway by being metabolized by a variety of cytochrome P450 enzymes. Okay, so these enzymes that are shown right here, let me do this in red. These enzymes right here, these are all cytochrome P450 enzymes. And of course, they have the typical catalytic cycle for P450s. And I highly recommend that you go watch the video on the P450 catalytic cycle so you can understand what they're doing. But effectively, what P450s do in this case and many other cases is they perform hydroxylation reactions. So if you look at this carbon right here on acetone that I'm highlighting in red, the P450 enzymes 2A6, 2E1, 4X1, 2S1, and 2U1 are going to hydroxylate that position. Okay, and just so you know, in case you haven't seen it up to this point, there are lots of P450 enzymes. Okay, there are basically too many to count. There are all sorts of varieties with various specificities, but it turns out that they all have broad specificity, And these are some of them that have the broadest specificity. And these are 5 of them that can react with acetone. And specifically, they're going to add this hydroxyl group onto acetone, and they generate this molecule, which is essentially called acetol. Okay, the "all" in the name is derived from the fact that it has an alcohol now. Okay, so if you look at these carbon number 1, 2, and 3, position 1 now has an alcohol. Okay, so this is called acetol. Okay, now here's an important distinction to understand. Okay, acetol, one of the P450s that creates it from acetone is called CYP2E1. Okay, so let me circle that in orange. This is CYP2E1. Okay, CYP2E1 also has a second activity. Okay, and that's denoted right here. The second activity is actually to use NADP+, and it's going to oxidize this alcohol into an aldehyde, as shown right here. So if CYP2E1, which is a P450, if that gets a hold of acetone, it's not just going to do this simple hydroxylation, it's also going to oxidize the alcohol into an aldehyde. Okay, so keep in mind that aldehydes are one oxidation step up from a hydroxyl group. So the key is CYP2E1 will not just hydroxylate, it'll oxidize the hydroxyl group into an aldehyde, and that generates something called methylglyoxal. And the "al" in the name is derived from the fact that it is an aldehyde. Okay, and it's specifically a 3-carbon aldehyde. The carbon numbering is exactly the same as it was in acetol. Okay, but there's another enzyme that can do this transformation of acetol to methylglyoxal. And that's, under the assumption that maybe one of these other P450 enzyme gets a hold of acetone. And what acetol can react with is something called methylglyoxal reductase. This is a type of aldose reductase. Okay, and aldose is essentially a carbohydrate resembling molecule that has an aldehyde, thus the name "al" in the aldose. And as the enzyme is named, you would think it would reduce the aldehyde into a hydroxyl group, but it turns out that these reactions are reversible. So this is a reversible reaction. And so, at least in the direction of acetol, or we could say acetone metabolism, okay, it runs from left to right. So technically this is an equilibrium reaction, okay? It's an equilibrium reaction, but when you load the system up with acetone, you're likely going to load the system up with acetol, and by Le Chatelier's principle, it will force the production of this, which is methylglyoxal. Okay, so it is an equilibrium reaction, but when you load the system up with acetol, it's going to force the reaction to the right. Okay, so now we have this methylglyoxal, and we're going to do something called the glyoxylase pathway. Okay, the glyoxylase pathway is going to utilize something called glutathione. Okay, so this molecule right here, this is called glutathione. Okay. In fact, this glyoxylase 1 that's shown right here has another name. Let me actually put it down here. The other name for this is called lactoyl glutathione lyase. So it is a class 4 enzyme, although when you first look at it, it doesn't really look like it so much, but it is. So this is lactoyl glutathione lyase, or glyoxylase 1. And it's going to use glutathione, and it's essentially going to take this 3-carbon fragment, which is methylglyoxal, and it's going to essentially attach it right here to glutathione. So now you have essentially this 1, 2, and 3 carbons there, but now this huge moiety, which I'll do in green, this huge moiety that's out here, all this business is glutathione. Okay. Now what's going to happen now is you get this lactoyl glutathione, so that's what this is. This is lactoyl glutathione. And then you're going to have lactoyl glutathione hydrolase, or also called glyoxylase 2. And it is a hydrolase, thus note the water here, and it's essentially going to hydrolyze off this business right here. Now when you look at that hydrolysis product, what you're essentially doing is you're splitting this bond right here. So this bond between this carbonyl and the sulfur, that's a thioester bond, but you're splitting it. Okay, so what you should get is this. And this right here, this is lactate. Specifically, it's R-lactate. This is R-lactate. So this carbon right here has R stereochemistry, and that is highly unusual. And you know anything about the normal lactate dehydrogenase, you would know that this stereochemistry is opposite. The lactate that's normally present, whenever you go out and run as fast as you can down the street and you start building up that lactic acid or lactate, that's S-lactate. So this has the opposite stereochemistry. But we're also going to react it with a lactate dehydrogenase, but this one is mechanistically unique. Okay, if we looked at the other lactate dehydrogenase, we would get a reaction that looked like this. It'd be very similar, where we would have a carboxylate, but instead what would happen is the hydroxyl group would have been going the opposite direction, right? And whenever we do what we abbreviate as LDH, lactate dehydrogenase, this is going to use NAD plus, you're going to get out NADH, and then you're going to get pyruvate, which is an alpha-keto acid. So there's pyruvate. And this is the S-lactate dehydrogenase. So that's an important distinction. That's S-lactate dehydrogenase. Okay? But this is a different one because we have the opposite stereochemistry. This is going to be R-lactate dehydrogenase. Okay? So if the cell has lots of lactate floating around, it's likely the S isomer. Okay? But through this pathway, you do generate R-lactate. Now, what you do is, instead of using NAD as an electron acceptor, you're going to use this guy right here. This is Q-oxidized, but another name for it is ubiquinone. Ubiquinone is an electron transfer protein in the inner membrane of the mitochondria. So that begs the question: if that's a coenzyme, an electron transfer coenzyme in the inner membrane of the mitochondria, is that where this enzyme is? And it turns out that that is in fact the case. Our lactate dehydrogenase is in a separate compartment than the other lactate dehydrogenase that we're used to hearing about. This is an enzyme that exists in the inner membrane. So this is the inner membrane of the mitochondria. Okay? And what it will do is it will take the electrons from our lactate. They will go into this, which is ubiquinone, to make ubiquinol. So this right here is ubiquinol. And then you end up with this, which is pyruvate. So this molecule, let me do this in a bold color. This is pyruvate, something that we're used to seeing. All this business back here, we may be very unused to. We may have never seen anything like this. But now we're in something that we're familiar with. We're familiar with pyruvate, and we know exactly what happens to it. Okay, if you're a cell that has intaken acetoacetate or acetone, you're likely a peripheral cell that is essentially running on ketone bodies. Okay, so what's going to happen is if you're that peripheral cell that needs energy, you're going to take this pyruvate and run it through the pyruvate dehydrogenase complex. Okay, and when you do this, you're going to get out an NADH, so that adds to the energy yield, and you're also going to get an acetyl-CoA. And the acetyl-CoA, as we know, goes into the TCA cycle. Now let's think about that acetyl-CoA as it goes into the TCA cycle. Well, as it goes into the TCA cycle, right, so we have this cycle right here, okay, where this is citrate synthase. Let's think about what you get. Well, you get out an NADH, and that's through the action of isocitrate dehydrogenase. The alpha-ketoglutarate dehydrogenase complex also gives you an NADH. If we go over to succinate dehydrogenase, that will give you an FADH2, but that transfers its electrons directly to ubiquinone, and that gets you reduced ubiquinol. And then from malate dehydrogenase, you also get an NADH. So let's really think about our yield here. Okay, yeah, we got an NADH from the pyruvate dehydrogenase complex, but really and truly, we really didn't lose anything. I mean, we gained a ubiquinol here. Let's keep track of that. We gained a ubiquinol. Let's see. We gained a ubiquinol. We also gained an NADPH through this reductase that runs from the direction of left to right. Okay, and I guess technically that NADPH would cancel with the one that was used for this P450 reaction, but overall, this is a tremendous yield, because not only did we get an NADH from the pyruvate dehydrogenase complex, but when that acetyl-CoA goes into the TCA cycle, we're going to get one NADH from isocitrate dehydrogenase, another one from alpha-ketoglutarate dehydrogenase complex, a reduced CoQ, or ubiquinol, from succinate dehydrogenase, and our final NADH from malate dehydrogenase. And if you really think about this, we also got a nucleoside triphosphate from succinyl-CoA synthetase. So I forgot about that as well. And that's going to be either where the nitrogenous base is guanine or adenine, which gives us either a GTP or an ATP. But overall, the function is going to be similar because these two can interconvert through nucleoside diphosphate kinase, which is another sort of nitrogenous-based equilibrium enzyme. Okay? But in any case, we've now seen the total catabolic pathway for acetone, and as we saw, you get a tremendous amount of energy from one catabolism of acetone. So let's do a quick recap. Acetoacetate will theoretically convert to acetone, and we know that to be the case because experimental evidence shows that. But whether or not this enzyme here exists is unknown. It hasn't been isolated, but the reaction in humans may occur spontaneously. Acetone will get hydroxylated by various P450 enzymes to acetol, and then acetol will get dehydrogenated to methylglyoxal, either through the latter action of site 2E1 or from methylglyoxal reductase, which is an aldose reductase that through Le Chatelier's principle runs toward the right. Then once we get methylglyoxal, we run it through the glyoxylase pathway. The first enzyme uses glutathione, and it's catalyzed by glyoxylase 1 to give us lactoyl glutathione, which is shown right here. And then glyoxylase 2 hydrolyzes off the lactate, but in this case, it's the R stereochemistry. And then this particular lactate ends up in an R-lactate dehydrogenase enzyme, but this enzyme doesn't use the NAD that we're used to seeing. It uses an oxidized coenzyme Q, or ubiquinone, picks up the electrons to make pyruvate, as shown here, and then you get totally reduced to ubiquinol in the process. So here we're not even considering this S-lactate dehydrogenase at all. And then the pyruvate, assuming we're a peripheral tissue where this stuff would take place, pyruvate gets converted into NADH and acetyl-CoA through the action of the pyruvate dehydrogenase complex. The acetyl-CoA goes into the TCA cycle, and overall, we get a tremendous yield per acetone. Hope this video helped. See you in the next video.